good evening, everybody. Welcome to this evening episode of Pursue. This is Pursue 19B, which is Hematology, Hemostasis, and Thrombosis. We are streaming live from IPGME&R Kolkata. We are here with a very basic topic, which is molecular biology and biochemistry of the coagulation factors and pathways of hemostasis, control of coagulation reaction, and vascular function in hemostasis. And to talk on that, we have Dr. Anindita Mondal, who's a very bright MD person who's done MBBS and MD in pathology. Presently, she's a senior resident at the IPGME&R and S.S.K.M Hospital, Kolkata, with areas of interest in oncopathology, histopathology, and cytopathology. Before I ask Dr. Anindita to start, let me request all of you to keep your mic muted, your camera off, and please do not share your screen. With this, let me request Dr. Anindita, ma'am, please share your screen. Let us start.
Thank you, sir, for this kind introduction. It's a great privilege and an opportunity to share my knowledge on this beautiful platform. So, with this few words and thank you, sir, again, I want to start my presentation.
Yeah, just share your PowerPoint.
Uh, so, it's visible, right?
Yeah, it's perfectly visible. Just press that hide thing so that that pop-up goes away. Yeah, must make it full screen.
Yes, sir.
Yeah, fine. Please start.
Thank you, sir. Today, my topic is molecular biology and biochemistry of coagulation factors and the pathways of hemostasis, control of coagulation reactions, and vascular function in hemostasis. Now, I'll discuss this topic under this following headings. So, the PPT is not moving. Okay.
Hello?
Yeah. Uh, Yes, sir. It's now okay. Molecular
Go ahead.
biology and biochemistry of the coagulation factors, pathways of hemostasis, coagulation cascade, factors that limit the coagulation, the fibrinolytic system, vascular functions in hemostasis, and finally the summary. This table shows the coagulation factors along with their respective names. Factor 1 fibrinogen, 2 prothrombin, 3 tissue factor, factor 4 is calcium, factor 5 is labile factor, factor 6 not present, factor 7 is proconvertin or stable factor, factor 8 anti-hemophilic factor or anti-hemophilic globulin, factor 9 Christmas factor, factor 10 Swart power factor, factor 11 plasma thromboplastin antecedent, factor 12 hegemon or contact factor, factor 13 the fibrin stabilizing factor also known as Lackey-Lorand factor. Apart from this, there are prekallikrein or the Fletcher factor and high molecular weight kininogen, the Fitzgerald factor. This table shows the factors along with the plasma half-life and the plasma concentration. Moving to the next slide. Now, what deficiency occurs in absence of this particular coagulation factors? Fibrinogen deficiency causes afibrinogenemia. Factor 5 deficiency produces parahemophilia. Factor 8 deficiency produces classical hemophilia or hemophilia A. Factor 9 causes hemophilia B or the Christmas disease. And factor 11, it causes hemophilia C. Von Willebrand factor deficiency leads to von Willebrand disease. This is a table which shows the molecular biology and biochemistry at a glance. It shows the genetic symbol, genetic location, the exons, the amino acids nature, the plasma level, the plasma half-life, and the main action of all these factors. This is the extended table. It shows the similar features of all those factors. Now, coming to the coagulation cascade. The coagulation cascade, it consists of a complex network of reactions for the conversion of zymogen cofactors into cofactors. The coagulation factors can be grouped as the vitamin K dependent zymogens, procoagulant cofactors, soluble cofactors, cell associated cofactors, fibrin network, and the inhibitors of coagulation. Now coming to the vitamin K dependent zymogens. What are those? These are prothrombin, factor 7, factor 9, factor 10 and protein C. The vitamin K dependent zymogens, they circulate in an inactive state and they require proteolytic activation to function as a serine protease. They all share a very similar structure with a C-terminal serine protease domain and an N-terminal gamma carboxy glutamic acid domain. This serine protease domain and the gamma carboxy glutamic acid domain, these two domains are interconnected by Kriegel domains. Here we can see Kriegel 1 and Kriegel 2 or epidermal growth factor like domains. Each protein has a defined function and they help in facilitating the substrate recognition, interaction with the protein cofactors, and to bind to the negatively charged lipid surface such as the activated platelets and the endothelial cells. All this results in restricting the coagulation to the site of injury. That is what is very important. Otherwise, there will be an uncontrolled coagulation. Now, this diagram, it depicts the vitamin K dependent zymogen. Here in this diagram, each small circle that represents an amino acid. The pre-pro leader sequence consists of the signal peptide as well as the propeptide, which directs the gamma carboxylation of the glutamic acid residues. Cleavage between this pre-pro sequence and indicates from the mature protein is indicated by this cleavage. The glutamic acid domains are indicated by this blue dots, by this blue circles. Prothrombin, this is, you see, this is the prothrombin. It has got a finger lobe which is followed by two Kriegel domains. These are two Kriegel domains, factor 7, 9, 10 and protein C. They have epidermal growth factor 1 and 2 like domains. Factor 10 and protein C, they circulate as a single chain molecules. Factor 10 and protein C, they circulate as two chains. Or after the proteolytic activation, all the molecules of the two chains are disulfide linked. This is the disulfide linked molecule which is indicated by this green arrow. Now this yellow region, this is the activated catalytic region. You see in this factor 9, 10 and the protein C. Now this schematic diagram, it shows the vitamin K dependent carboxylation. The vitamin K dependent carboxylation is very essential. This carboxylation requires oxygen, carbon dioxide and reduced form of vitamin K for its action. Hence vitamin K dependent, hence these are known as the vitamin K dependent proteins. For one molecule of glutamic residue that is carboxylated, one molecule of reduced vitamin K is converted to epioxide form. This is done by the carboxylase. Now this vitamin K epioxide is again converted to reduced form by epioxide reductase. Warfarin and other 4-hydroxycoumarine related compounds, they inhibit this epioxide reductase. Because warfarin, it blocks this epioxide reductase and not the carboxylase. This inhibition caused by warfarin and other 4-hydroxycoumarine compounds, these are temporary, which is overcome by administration of vitamin K. This picture shows the calcium dependent association of the glutamic acid domain with the anionic phospholipid surface. Under normal conditions, what happens? Phosphatidylserine is not exposed to the outer membrane leaflet of the cells. In activated endothelial cells and the platelets, this phosphatidylserine is a part of extracellular cell surface where it supports the coagulation system. The glutamic acid domain, which is represented by this red circles, interacts with the anionic cell surface in a calcium dependent manner. The calcium ions are coordinated by the GLA residues and they cause a hydrophobic surface loop. Membrane binding by the glutamic acid domain occurs when this hydrophobic surface loop they penetrates into the hydrophobic portion of the phospholipid bilayer. Glutamic acid bound calcium ions with the phosphate head groups of the phosphatide binds with the phosphate head groups of the phosphatidylserine. Now coming to the next group, the procoagulant cofactors, which are 5 and 8. Factor 5 and factor 5 and factor 8 both function as cofactors in coagulation and they dramatically enhance the catalytic activity of the macromolecular complexes, resulting in generation of the thrombin and factor 10A. This shows a schematic diagram of factor 5 with A1, A2, B, A3, C1 and C2. The thrombin cleavage sites are indicated by these green arrows. The 709, arginine 1018 and arginine 15145. Whereas the activated protein C cleavage sites are indicated by these red arrows. Factor 5 is a non-enzymatic cofactor within the prothrombinase complex that greatly accelerates the ability of factor 10A to convert prothrombin to thrombin. Activated protein C catalyzes the inactivation of factor 5A by cleavages at the main sites, arginine 306, arginine 506, upon which the cleaved A2 fragment dissociates from the factor 5A and factor 5A can then no longer associate with factor 10A. Arginine 506 GLN mutation in factor 5 leads to a resistance to inactivation by activated protein C. This is called factor 5 Leiden mutation and is associated with an increased risk of venous thromboembolism. Homozygosity or compound heterozygosity for the loss of function mutations in factor 5 leads to a bleeding disorder which is known as parahemophilia or Aurin parahemophilia. In whites, most of gain of function mutation in factor 5 is known as factor 5 Leiden, which leads to plasma abnormality known as activated protein C resistance. Factor 7. The schematic diagram is shown below. It's represented by A1, small a1, A2, A2, B, A3, A3 and C1, C2 domain, where the acidic regions are denoted by small a1, a2, a3. Thrombin cleavage sites are arginine 372, 740 and 1689 indicated by these green arrows, and activated protein C cleavage sites are indicated by these red arrows. Thrombin and factor 10A are principal activators of procofactor 8 and generate the cofactor 8A through the sequential proteolysis at arginine 740, arginine 372 and arginine 1689. After being activated, factor 8A function as cofactor for factor 9A in phospholipid dependent conversion of factor 10 to factor 10A. As we know, a defect or deficiency in factor 8A leads to hemophilia A. High level of factor 8 are common and a strong risk factor for venous thrombosis. Coming to the soluble cofactors, protein S and the von Willebrand factor. The protein structure of protein S differs from other vitamin K dependent proteins. Free protein S, it serves as a cofactor for activated protein C in the proteolytic inactivation of factors 5A, 8A. Loss of function mutations in the PROS1 lead to protein S deficiency. Heterozygous deficiencies of protein A can be categorized into three types of deficiency. Type 1 deficiency is characterized by antigen levels which are less than 50% of the normal. In type 2, the antigen levels are near normal, whereas the activity of this antigen are decreased by 50%. In type 3, there is a low level of free protein S. In keeping with this classification, the clinical laboratories offer a protein A activity assay, free antigen assay, or total antigen assay, as the condition may be. Now, this schematic diagram shows the protein S, factor 11, factor 12, and TFPI, tissue factor pathway inhibitor representation. The structure of protein S differs from other vitamin K dependent proteins because it lacks a serine protease domain and consequently there is no catalytic activity. Protein S is composed of GLA domain comprising of 11 glutamic acid residues, a thrombin sensitive region, this region, epidermal growth factor domains, and a C-terminal sex hormone binding domain. Protein S, factor 11 and factor 12 and TFPI, tissue factor pathway inhibitor, but protein S, coming to the next slide, I'll discuss factor 11, 12 and TFPI later on. The von Willebrand factor, it's a large multimeric protein which is required for normal platelet adhesion to the component of the blood vessel, and it acts as a carrier for factor 8. It is synthesized in the megakaryocytes and endothelial cells and stored in specialized organelles in platelets and endothelial cells. Large von Willebrand factors multimers are cleaved by plasma protease ADAMTS13, which is a disintegrin, and a metalloproteinase with thrombospondin motifs 13. This cleavage produces smaller von Willebrand factor multimers which circulate in the plasma. Reduced ADAMTS13 activity is linked with various microangiopathies with increased platelet activity. Large von Willebrand factor multimers are more active than the smaller ones. Qualitative or quantitative deficiencies in von Willebrand factor cause von Willebrand disease, a mild to severe bleeding disorder. Quantitative deficiency of von Willebrand factor leads to type 1, type 3 von Willebrand disease, and functional defect, it leads to type 2 von Willebrand disease. Type 1 von Willebrand is the most common form, whereas type 3 is the most severe. Now, we all know deficiency of factor 8, it causes hemophilia. But a function of von Willebrand factor is to carry the factor 8. Along with von Willebrand factor, the half-life of factor 8 is about 12 hours. But what happens if von Willebrand factor is absent? Then factor 8 is degraded very soon. It's almost degraded in 2.5 hours. So, this behaves as if there is factor 8 deficiency. It's known as pseudo-hemophilia. Factor 9 and the contact system. Factor 11 and the contact system. After the activation of the factor 11, binding sites for the substrate of factor 9 become available in the APL3 domain. Now, let us move. This is factor 11. The factor 11, it has got this catalytic domain, and these are the APL domain. APL domain 1, 2, 3 and 4. and the serine protease domain of the factor 11. Factor 11 proteolytically activates factor 9 to form factor 9 activated factor 9A in a calcium dependent but phospholipid independent manner. Both forms of factor 11A dimer as well as monomeric factor 11A activate factor 9 in a similar manner. Factor 12A dependent activation of factor 11 is not essential for normal hemostasis, but this is what it happens in pathological thrombus formation. Thrombin mediated activation of factor 11, on the other hand, seems most significant under conditions of low tissue factor and is assumed to enhance clot stability through thrombin activation of thrombin activable fibrinolysis inhibitor. Factor 12 deficiency is not associated with bleeding. Factor 12, on the other hand, contributes to surface-induced pathologic thrombosis via activation of factor 11. The SARPIN's C1 inhibitor is the main plasma inhibitor of alpha factor 12A and beta factor 12A. In addition, antithrombin and plasminogen activator inhibitor also inhibit factor 12A activity. Conditions in which factor 12A activity is not properly controlled, such as in C1q inhibitor deficiency states or in case of constitutively active form of factor 12A, can result in a disorder, hereditary angioedema. Coming to the cell-associated cofactors, tissue factor, thrombodilin, endothelial protein C receptor. Tissue factor, also known as thromboplastin or CD142, is the cellular receptor and cofactor for factor 7 and 7A. Tissue factor, activated factor 7A complex is the major physiologic initiator of blood coagulation. Coagulation is initiated when an injury occurs in a vessel and allows blood to come into contact with extravascular tissue factor. Escape of blood from the vessel allows factor 7 to bind to extravascular tissue factor and initiate coagulation following proteolytic activation of factor 7 to activated factor 7A. An injury in the extravascular tissue factor 7A complexes to come into contact with blood and initiate thrombin generation and activated on the activated platelet surfaces. Interaction of tissue factor with activated factor 7A induces conformational changes in the serine protease domain of factor 7A, allowing the latter to proteolytically activate factor 9 and factor 10. This is the schematic diagram showing the tissue factor and the factor 7A complex. Here we can see the catalytic domain, epidermal growth factor 2 and 1 domain and the glutamic acid domain. This is the tissue factor, binding of this tissue factor with the factor 7A. It changes the conformation, it changes the shape of factor 7 and also opens the active site. The fibrin network, the fibrinogen, factor 13 and thrombin actifiable fibrinolysis inhibitor. Fibrinogen, when converted to fibrin, forms the structural meshwork that consolidates an initial platelet plug into a solid hemostatic plug. This schematic diagram shows the structure of fibrinogen. Fibrinogen is a dimer. These are two identical units. Each monomer, it consists of 3 chains. These are represented by alpha, beta and gamma. These are color coded. This alpha is shown by this light blue, beta is shown by this pinkish and gamma is shown by this deep blue color. The disulfides that link the two monomers are indicated in the central E domain. The D domains consist primarily of the C terminal of the B beta and gamma chains. The helical region connecting the two domains consists of all the three chains intertwined. Now thrombin, it binds to the central domain of the fibrinogen and proteolytically releases two fibrinopeptides, alpha residues 1 to 16 and two fibrinopeptides B beta residues 1 to 14 from each fibrinogen molecule. Release of the fibrinopeptides exposes the binding sites in the E domain that have complementary sites in the D domain of other fibrin monomers. These complementary binding sites lead to initial formation of two-stranded protofibril. These are the two-stranded protofibril with half staggered overlap configuration. As you can see in this diagram, protofibrils then aggregate into thick fibers that branch into a meshwork of interconnected thick fibers. So this structure is indicated schematically in this diagram. Now coming to factor 13. The glycoprotein factor 13 is a pro-transglutaminase that upon activation cross-links and stabilizes the fibrin clots. Factor 13 activation, the structure of factor 13A and B subunits, these are the factor 13A and the B subunits are indicated schematically. This A are indicated in the red and the B are indicated in the green. Cleavage to this activation peptides of factor 13A by thrombin releases the factor 13B subunits, the B chains, and induces a conformational changes in the factor 13A that opens up the active site. Now, let us move to the next part of today's discussion, that is hemostasis. What is hemostasis? Hemostasis is a precisely orchestrated process which involves platelets, clotting factors, endothelium, which occurs at the site of vascular injury and culminates in the formation of a blood clot, which serves to prevent or limit the extent of bleeding. What are the sequence of events that lead to hemostasis at a site of vascular injury? Arteriolar vasoconstriction, primary hemostasis, formation of the platelet plug, secondary hemostasis, deposition of fibrin, clot stabilization and resorption. We will go through all this one by one. Vasoconstriction. Arteriolar vasoconstriction occurs immediately and it reduces the blood flow to this injured area. This vasoconstriction is mediated mainly by reflex neurogenic mechanisms, which is augmented by local secretion of factors such as endothelin, which is a potent vasoconstrictor. This effect is, however, transient, and the bleeding will continue if nothing further happens. Now comes into play platelet activation and aggregation. So this vascular injury, it exposes the von Willebrand factor, this red dots you can see, and the subendothelial collagen. What happens? Von Willebrand factor and the subendothelial collagen, they causes platelet adherence and activation. What is meant by activation? Activation, it results in the dramatic shape change. The platelets, which are small round discs with spiky protrusion, rounded discs, they become flat plates with spike-like protrusions, which increases the surface area and causes release of these granules. So we'll later come to see the granules which are present in platelets. At this moment, there is activation of the platelets followed by shape change and the granule release. Within a few minutes, what happens? Additional platelets undergo aggregation to form a primary hemostatic plugs. When the platelets change the shape, there is accompanying alterations in the GP2B3A, which increases the affinity for fibrinogen, and by translocation of the negatively charged phospholipid to the platelet surface. Platelet activation is triggered by a number of factors, including the coagulation factor, thrombin and ADP. Thrombin activates the platelets through a special G protein coupled receptor known as the protease activated receptor, which is switched on to a proteolytic cleavage carried out by thrombin. So here it forms the primary hemostatic plug. Now, as I was talking, let us have a glance of the ultrastructure of the platelets. This is the ultrastructure of the platelets depicting the glycocalyx, the plasma membrane, the open canalicular system, submembranous filaments or the platelet contractile proteins, the dense tubular system, mitochondria, the glycogen. There are two types of granules, the alpha and the dense granules. The dense granule, it contains the ADP, calcium, serotonin, and the specific alpha granule, it contains fibrinogen, factor V, von Willebrand factor, fibronectin, heparin antagonist, platelet factor 4, PDGF and other proteins. Now the platelet aggregation occurs following their activation. The conformational change in the glycoprotein 2B3A, this semicircle, a dark blue semicircle GP2B3A that occurs with platelet activation allows binding of the fibrinogen, a large bivalent plasma polypeptide, leading to their aggregation. Now, in cases where this GP2B3A is absent, it leads to Glanzmann thrombasthenia. The initial wave of aggregation is reversible, but with concurrent activation of thrombin stabilizes the platelet plug and causes further activation and aggregation. When this GP1B receptor is absent, it produces the Barnard-Solier and von Willebrand factor. If it is deficient, it produces von Willebrand disease. This shows platelet adhesion. This process is called adhesion when the platelet binds with the endothelial. Von Willebrand factor functions as the adhesion bridge, and this is aggregation between the two platelets, which occurs due to this GP2B3A complex in presence of fibrinogen. Activation of clotting factors and formation of fibrin. Vascular injury also results in the release of tissue factor. What is tissue factor? Tissue factor is a membrane-bound procoagulant protein which is normally expressed by the subendothelial cells in the vessel walls, smooth muscles and the fibroblasts. Tissue factor, it activates factor 7, which sets the motion of the cascade of reaction which produces thrombin. Thrombin cleaves circulating fibrinogen into insoluble fibrin, creating a meshwork of fibrin, and is also a potent activator of platelets. It causes additional platelet aggregation. This entire thing, this is called the secondary hemostasis, which occurs after the primary initial platelet plug. Now the platelet plug is formed, the secondary hemostatic plug is formed. Now this polymerized fibrin and platelet aggregates, they undergo contraction to form a solid permanent plug, which prevents any further hemorrhage. This point is crucial. Counter-regulatory mechanisms such as tissue plasminogen activator, which is produced by the endothelial cells, they prevent the further spreading of this clot, and the clot is limited to this site of injury, and this also causes the clot resorption. So this entire thing which we have just talked about, this is shown schematically in this diagram. This is the vessel injury which causes the platelet exposure of the collagen, causes platelet adhesion. Also it causes tissue factor release and vasoconstriction. This platelet adhesion, it promotes platelet activation by shape change, granule secretion and activation of the GP2B3A. This platelet activation causes serotonin release, which further produces vasoconstriction. It causes release of thromboxane ADP, which again promotes vasoconstriction, and the platelet phospholipid, they participate in the blood coagulation cascade. This platelet activation in presence of thromboxane ADP, it promotes the platelet aggregation, and vasoconstriction also reduces blood flow. It helps in the formation of primary hemostatic plug, which is further stabilized by thrombin, and fibrin participates to form the stable hemostatic plug. So the coagulation cascade, what is it? It's a series of amplifying events, enzymatic reactions, which leads to the deposition of an insoluble fibrin clot. Each reaction in this involves an enzyme, an activated coagulation factor, a substrate, an inactive proenzyme form of the coagulation factor, and a cofactor, which is a reaction accelerator. These components are assembled on the negatively charged phospholipid surface, which is provided by the platelets. Assembly of the reaction complexes depends on calcium, which binds to the gamma carboxylated glutamic acid residues that are present in factor 2, 7, 9 and 10. The enzymatic reactions that produce the gamma carboxylated glutamic acid use vitamin K as a cofactor, and which are antagonized by drugs such as coumadin. This flow diagram, it shows the coagulation cascade in the laboratory and in vivo. In the laboratory, what happens? The intrinsic pathway is activated by the negatively charged surface, example the glass beads. It converts factor 12 to activated factor 12A. Factor 11 is then converted to factor 11A in presence of activated factor 12A. Factor 9 is converted to 9A in presence of activated factor 11A. This 9A and the activated factor 8A, this is known as intrinsic tenase because it converts factor 10 to activated factor 10A, which converts prothrombin to thrombin. Thrombin converts fibrinogen into fibrin clot. In the meantime, tissue factor is also activated. Tissue factor also binds with the factor 7 to form this tissue factor activated factor 7A complex, which is known as the extrinsic tenase. It activates 10 and factor 10, and this presence of activated factor 5, this converts prothrombin to thrombin. Whereas in vivo, the scenario is a bit different. In vivo, it's mostly by this extrinsic pathway where factor 7 is converted into activated factor 7A in presence of tissue factor. Then tissue factor, factor 7A, activated factor 7A complex converts factor 9 to factor 9A. Activated factor 9A in presence of activated factor 8A converts 10 to 10A, and then further in presence of activated factor 5A it converts prothrombin to thrombin, which converts fibrinogen to fibrin clot. This is the same diagram which is shown schematically over here. Phospholipid and calcium are required as cofactor in these steps, and thrombin activates these steps. Now, how do you measure this coagulation pathways? The intrinsic pathway and this common pathway, it is measured by factor. It is measured by APTT, activated partial thromboplastin time. Whereas this extrinsic pathway and this common pathway is measured by prothrombin time. To measure thus how to measure this APTT? It is checked by putting negative charge in collect the sample in a glass tube and add negative charge. How this negative charge is added? This negative charge is added by putting kaolin, silica, etc. Coagulation study should not be carried out, this coagulation test should not be carried out in a glass tube, because glass is already providing the negative charge. Also, this APTT, it is given in seconds, so it should not be given in isolation. It should be always provided with a control. The control is obtained from the pooled sample. The normal blood donor who come to the blood bank, and the control should be done from that. How sensitive is APTT? APTT can detect coagulation factor deficiency when the coagulation factor deficiency fall at least 15 to 20% of the normal. What factors does it detect? It detects factor 12, 11, 9, 8, 10, 5, 2 and 1. The measurement of this extrinsic pathway, how to check the extrinsic pathway? We add tissue factor to the sample. The source of tissue factor is human brain or rabbit brain. Then, but the quality here differs, because obviously human brain gives a better quality than to rabbit brain. So there was a disparity between the results. For this, the WHO, World Health Organization, came with the International Standardized Index. The tissue factor which is manufactured by any company, it should be compared to the tissue factor which is present in the WHO. More the value is towards 1, the tissue factor is more better. But we have to compare the tissue factor with the control values. So it was proposed that the international normalized ratio or INR is put into effect. What is INR? It is the PT of the test by the PT value of the control, which is raised to the power of ISI. INR should be maintained in the therapeutic range. INR for the treatment of DVT should be maintained between 2.5 to 3.5. For mechanical heart valves, it should be between 2 to 3 for both prophylactic and therapeutic use of DVT. Now coming to the screening test for hemostasis. What are the tests we can perform to check the primary hemostasis? Bleeding time, the platelet and the vascular phages, PFA 100 system to assess the platelet function, platelet count to detect the quantization of the platelets, blood smear, quantitative and morphologic abnormalities, and detection of underlying hematological disorder. What are the tests for secondary hemostasis? Clotting time, which is the crude test for coagulation pathways, prothrombin time, the extrinsic and the common pathways, and the activated partial thromboplastin time to test the intrinsic and the common pathways. This table shows the screening tests used in the diagnosis of the coagulation disorders. Thrombin time, it detects the deficiency or abnormality of the fibrinogen or inhibition of thrombin by heparin or fibrin degradation products. It is raised mostly in DIC, heparin therapy. Prothrombin time, deficiency or inhibition of one or more of the coagulation factors 7, 10, 5, 2 and fibrinogen. It is raised in liver disease, warfarin therapy and DIC. Activated partial thromboplastin time or the partial thromboplastin time, deficiency or inhibition of one or more of the following coagulation factors 12, 11, 9, Christmas disease, factor A deficiency, hemophilia, 10, 5, 2 and fibrinogen. It is raised in hemophilia, Christmas disease plus the above conditions. Fibrinogen quantification, it is characterized by fibrinogen deficiency raised in DIC and liver disease. Now, what is the role of thrombin? Thrombin is a very important in coagulation system because it has various enzymatic activities and control diverse aspects of hemostasis, and also it links to clotting, to inflammation and repair. Among the most important activities of thrombin are the conversion of fibrinogen into cross-linked fibrin, which I have seen, platelet activation, pro-inflammatory effects and anticoagulant effects. Conversion of fibrinogen into cross-linked fibrin. Thrombin, it converts soluble fibrinogen into fibrinogen monomers that polymerize into an insoluble fibrin, and it amplifies coagulation process not only by activating factor 11 but by activating two main factors, cofactors, factor 7, factor 5 and factor 8. It also stabilizes secondary hemostatic plug by activating factor 13, which covalently cross-links fibrin. Platelet activation. Thrombin, as we know, is a potent activator of platelet activation and aggregation through its protease activated receptors, thereby linking platelet function to coagulation. Pro-inflammatory effects. The protease activated receptors are also expressed in inflammatory cells, endothelium and other cells, and activation of these receptors by thrombin is believed to mediate the pro-inflammatory effects which contribute to the tissue repair and angiogenesis. What are the anticoagulant effects? Thrombin also acts on the normal endothelium. It changes the endothelium from a procoagulant to anticoagulant state, which helps in reversal and prevent clotting beyond the desired site of the vascular injury. So this is pictographically represented thrombin. It produces the actions of thrombin. It produces platelet activation, inhibits fibrinolysis, activates factor 5 to factor 5A, activates factor 8 to factor 8A, converts fibrinogen to fibrin. It plays a role in inflammation, protein C activation, factor 11 activation, and factor 13 activation and stabilization. Here it shows how fibrinogen is converted in presence of thrombin into fibrin monomer, which further undergoes hydrogen bonding to form fibrin polymer, and then it undergoes transaminase bonding in the presence of activated factor 13A, which is again formed by in presence of thrombin from factor 13. So activated factor 13A, it transforms fibrin polymer into cross-linked fibrin. Role of thrombin in hemostasis and cellular activation. This is again pictorially represented. It causes platelet aggregation at the site of injury by release of thromboxane and fibrin deposition. These are the pro-inflammatory effects. These are the anticoagulant effects by release of nitric oxide, prostacyclin, and these are the inflammatory effects. Now coming to the factors that limit the coagulation system. Coagulation, once initiated, it has to be restricted to the site of vascular injury. Otherwise, it would be very disastrous. One of the limiting factors is simply the dilution of the blood. The blood flow increases, it takes away the coagulation factors, which is washed off by the liver. The second is the requirement of the negatively charged phospholipids, which as mentioned are provided by the platelets that have been activated by the contact with subendothelial matrix at the site of vascular injury. Last but not the least, the most important counter-regulatory mechanisms involves the factor that are expressed by intact endothelium itself adjacent to the site of injury. Herein comes into play the fibrinolytic system. Activation of the coagulation cascade also sets in the fibrinolytic cascade, which limits the clot size and later causes dissolution of the clot. Fibrinolysis is accomplished through enzymatic activity of plasmin, which breaks down fibrin and interferes with its polymerization. And elevated levels of products of fibrinogen, often called fibrin split products, most notably the D-dimers are a useful marker in several thrombotic states. Plasmin is generated by enzymatic catabolism of the inactive circulating plasminogen, either by factor 12 dependent pathway, possibly, and explains the role of association of factor 12 deficiency and thrombosis or plasminogen activators. The most important of which is tissue plasminogen activator. This tissue plasminogen activator, it is synthesized by the endothelium and is most active when it is bound to the fibrin. The tissue plasminogen activator is used as a therapeutic agent because of its fibrinolytic activity is confined to the area of recent thrombosis. Once activated, plasmin is in turn tightly controlled by counter-regulatory mechanisms such as alpha-2 antiplasmin inhibitor, alpha-2 plasmin inhibitor, and which is a plasma protein that binds and rapidly inhibits free plasmin. This is the schematic diagram showing the fibrinolytic system. Here, plasminogen is activated to plasmin in presence of intrinsic activation by factor 12A, kallikrein, extrinsic activation, tissue plasminogen activator, urokinase like A, and in presence of drugs such as streptokinase, this plasmin, it converts fibrin into fibrin degradation products. Let us have a look at the vascular functions. What role does endothelium has to play in hemostasis? We have seen a lot of role till now. Jotting it down, the endothelium, the balance between the anticoagulant and the procoagulant activities of the endothelium often determines whether the clot formation, propagation, or dissolution occurs. The antithrombotic properties of endothelium can be divided into activities directed at platelets, coagulation factors, and fibrinolysis. Platelet inhibitory effects. The intact endothelium serve as a barrier between the platelets from the subendothelial von Willebrand factor and collagen. It releases prostaglandin, nitric oxide, adenosine phosphatase, and bind and alter the activity of thrombin. The anticoagulant effects, normal endothelium, it shields the coagulation factors from tissue factor in the vessel wall and expresses multiple factors that actively oppose the coagulation, such as thrombodulin, endothelial protein C receptor, heparin-like molecules, and tissue factor pathway inhibitor. The fibrinolytic effects, normal endothelial cells synthesize tissue plasminogen activator, a key component of the fibrinolytic pathway. The anticoagulant properties of normal endothelium. It releases nitric oxide, PgI2, which inhibits the platelet aggregation. Endothelium, it releases tissue plasminogen activator, activates fibrinolysis. Heparin-like molecules on the surface bind and activate antithrombin 3. They activate the antithrombin 3, which inhibits the thrombin. And factors 9 and 10A. tissue factor pathway inhibitor inactivates tissue factor and activated 7A factor 7A complex. Thrombodilene it inhibits protein C to active protein C formation, and active protein C it inactivates factor 5A and 8A. This is the slide showing the procoagulant activity. Exposure of the membrane-bound tissue factor, it causes the extrinsic coagulation sequence, leads to the extrinsic coagulation sequence. Platelet adhesion is by fibrinogen is indicated, and this von Willebrand factor, which helps in adhesion of the platelet with this von Willebrand factor, helps to form further platelet aggregation. These are the procoagulant activities in the shown by the endothelium. To summarize, what we all have got to know till now, endothelial injury exposes the underlying basement membrane. Platelet adhere to the extracellular matrix primarily through the binding of the GP1B receptors to the von Willebrand factor. This process is known as adhesion. Adhesion, it leads to platelet activation, which is associated with secretion of the platelet granule contents, including calcium ADP. There is a dramatic change in the shape and membrane composition and activation of GP2B3A receptors. The GP2B3A receptors on the platelets form bridging crosslinks with fibrinogen, which leads to platelet aggregation. Concomitant activation of thrombin promotes fibrin deposition, cementing the platelet plug in place. Coagulation occurs via the sequential enzymatic conversion of the cascade of circulating and locally synthesized proteins. Tissue factor is the most important initiator of the coagulation cascade in vivo. At the final stage, thrombin converts fibrinogen into insoluble fibrin that contributes to the formation of the definitive hemostatic plug. Coagulation is restricted to the sites of vascular injury by limiting enzymatic activation to the phospholipid surfaces provided by the activated platelets or the endothelium. Circulating inhibitors of coagulation, such as antithrombin 3, whose activity is augmented by heparin-like molecules, is expressed on the endothelial cells. Expression of thrombodilene on normal endothelial cells, which bind thrombin and convert it to anticoagulant activity. Activation of fibrinolytic pathways by association of tissue plasminogen activator with fibrin. These are my references. And thank you. I end this with a beautiful picturesque Gurudong Mark Lake. Thank you so much.
Thank you, Dr. Anindita. Nice presentation. You were, I think, a bit nervous, that's all.
Yes, sir.
But otherwise, it went out very well, very smooth. And we were able to understand the entire cascade of coagulation, as well as the role of endothelium, as well as other factors of anticoagulation as well. Nice presentation. Thank you. We will invite you again for some other presentation as well in the near future. Right.
Thank you so much, sir.
Right. Take care. Bye-bye. Good night.
Bye.